Okay, so let's continue where we stopped last week. Remember, we were talking about the whole idea of metabolism as being a set of catabolic and anabolic reactions in the cell. And we were talking about oxidation and reduction. But one important thing about the chemical reactions in the cell is that they must go pretty fast. And in order to do so, cells utilize enzymes. So enzymes are biological catalysts. Most of the enzymes are proteins. And what they do is reducing the activation energy of the reaction. So if you look at the graph right here, it shows you the reaction path over time and the changes in energy. So when you start with the reactants and you transition to the products, it isn't just a smooth slide up. You have to go over that special hump, which is activation energy. So in the green color, you can see the activation energy for a reaction that does not have a catalyst, such as an enzyme. However, the addition of the catalyst reduces the activation energy, making that hump much smaller. Usually, compared to your attempt to throw a big boulder from the top of the building, if the railings are really high, you can't conceivably put the boulder over the railings. You just don't have enough physical strength. But if the barrier is pretty low, you can kind of push it through. Okay? So what I want to specifically emphasize is that the result of the reaction, the products, are the same. So effectively, enzymes, they increase the rate of the reaction, but they do not alter the yield or the ratio of the products. So what you get in the end is exactly the same as what you would get if you wouldn't use a catalyst. You just get into that final point much quicker. So how does that work? So the gray molecule is the enzyme, and the substrate, the chemical that enters the reaction, the reactant essentially, the substrate, binds into the active site of the enzyme. So now you can see the complex between the enzyme and the substrate. Then the structural features of the active site make the conversion of the substrate into products happen. We're not going to get into biophysics and biochemistry of this, but enzyme makes this conversion from this substrate to these two products much faster. And then products are released. What I really want you to appreciate is that if you look at the enzyme in the beginning and the enzyme at the end of the reaction, it doesn't change. So enzymes that facilitate certain reaction aren't wasted in these reactions, aren't destroyed, aren't modified. They come out of that reaction unaltered. Now, enzymes are extremely specific. Now, that being said, they can facilitate a very specific reaction But as we will see pretty soon, there is a chance to design molecules that would look similar to a substrate in order to inhibit the enzymatic function. But when I say that the enzymes are specific, what I mean is, if you have an enzyme that, I don't know, breaks down a milk sugar, lactose, it's going to break down the milk sugar. It's not going to break down sucrose or glucose. Well, glucose is monosaccharide, so it's not going to break down sucrose or maltose. It's only going to be lactose. Also, enzymes have optimal conditions for their function. So one thing that I want you to appreciate about these optimal conditions, if you try to build a graph of the enzyme activity versus, for example, pH, the enzyme activity will look like this. So there's an optimal pH, but the enzyme is still active outside. Its activity is just lower than it would be at its optimal pH. The same principle can be applied to temperature or concentration of different ions or whatever condition you can think of. And right now you might think that, this data is pretty relevant, but this data are relevant because if you think about bacterial enzymes working in optimal conditions, if you somehow can alter the conditions and can take bacteria out of the comfort zone, they won't be able to grow because the enzymes would not act the maximum capacity. Okay? So enzymes that bacterial cell produces can be secreted outside of the cell. In this case, we call them exoenzyme. The example of such an exoenzyme is a collagenase of Clostridium perfringens, the enzyme that bacteria release to break down collagen in the connective tissue. Another good example is an enzyme called coagulase. Coagulase is produced by Staphylococcus aureus. And when Staphylococcus is in the blood, coagulase causes clotting. Endoenzymes, to the contrary, stay within the cell. The good example would be something like RNA polymerase or enzymes that regulate protein synthesis, for instance. Enzymes also can be categorized as constitutive, regulated. Constitutive enzymes are produced at the same level, no matter what external environment brings in terms of changes. Regulated enzymes, are regulated. Their production is usually changed in response to changes in the external environment. So the activity of enzymes can be regulated through inhibition or through activation. So first, let's talk about inhibition. So here, you can see an enzyme with its active site occupied by the inhibitor. You can see that in this case, since active site is blocked, pretty much physically blocked, the actual substrate cannot bind to the active site and the enzymatic reaction cannot proceed. The alternative to competitive inhibition is a non-competitive process in which non-competitive inhibitor binds to a regulatory site, so-called allosteric site, and I will comment on what this word means, allosteric site, and that binding, as you can see, changes the conformation of an active site. So substrate cannot bind it again, but for a different reason, because it's just misshapen now. Well, the question that you should ask yourself all the time, how is that relevant? Turns out, a fair amount of drugs that we use to treat bacterial and viral infections are inhibitors of certain enzymes. Some of them are competitive inhibitors. Some of them are not. Some of them are non-competitive inhibitors. So you may ask yourself, why is that important to know if the inhibitor is competitive or not? I can give you an example. So this example is going to sound very weird on the recording, but you all can see what's going on. So Olivia and I, there is a chair between us. Let's say this chair is the active site. Okay? Olivia is a substrate, and I'm the inhibitor. So if I get to this chair first, competing with Olivia, right? I inhibit. Does that make sense? Now, here's my question, and that's going to sound silly, but hear me out. What if there are 10 Olivias and just one me? She gets more chances getting into that site. But if there's 100 me and just 10 Olivias, I have more chances. I don't know if you've heard about the antibiotic called Bactrim. Bactrim contains two competitive inhibitors of two enzymes that are essential for the production of the metabolite called folic acid in bacteria. And some bacteria develop resistance to that antibiotic by producing an enormous number of enzymes. So now imagine that we're not competing for the same chair, but we are, suddenly I have to block Olivia on 100 chairs. Chances are I'm not going to succeed. Okay? So it shows you both the mechanism for the antibiotic action and the way bacteria can evade these antibiotics. There are certain drugs that inhibit the reproduction of HIV that act as non-competitive inhibitors of certain enzymes. And in case of non-competitive inhibitor, you can imagine that if I'm a non-competitive inhibitor, that chair is changed in a way that no matter how many Olivias are out there, they can't get into this. So in some way, non-competitive inhibitor may be a better choice for a drug. So far, difference between non-competitive and competitive inhibition, I hope it is clear. So non-competitive inhibition, it can be also called allosteric. So the word allosteric refers to, okay, now I apologize, I don't know if it's Latin or Greek, most likely Latin, okay? Allo means different, and steric refers to the space, a different space. When the inhibitor binds not to the active site, but to some allosteric site, we call it allosteric inhibition. In the same way, molecules can exert allosteric activation by binding to some site at the protein, changing its shape so that active site becomes available for the substrate. The importance of this is not only because of various drugs, but also because of the regulation of metabolic pathways. A lot of intermediates of the metabolic pathways serve as allosteric inhibitors or activators of certain enzymes. Now, having said that, there is another concept that I want you to understand. It's the idea of the cofactor in the apoenzyme. So in this illustration, the gray molecule is the apoenzyme. It is not functional because its active site isn't complete yet. When an apoenzyme binds when an apoenzyme binds a cofactor or coenzyme, cofactors usually metal ions, coenzymes are organic molecules. When apoenzyme and cofactor coenzyme finally bind together, they form what's called a holoenzyme. And this holoenzyme now has the functional active site which can bind a substrate. So holoenzyme is actually active. Okay, sounds good? Now, last bit that I want to discuss over here is the idea of a pathway, the inhibition of a pathway. So what you're looking at here is the chain of enzymes that convert initial substrate through the series of the intermediates into the end product. So this linear pathway, as you can imagine, if you let it run without any restrictions, will deplete the cell of a substrate, whatever it is, and will generate excessive amount of the product. So how can we regulate that? And in this case, our helper is a negative feedback. When the end product accumulates, it may serve as the allosteric inhibitor, blocking the function of the very first enzyme in the pathway, and therefore stopping the entire enzymatic route. How is it relevant? Turns out that the activity of glycolytic enzymes, the enzymes that are first to break down glucose in the process of glycolysis, the activity is regulated by the ultimate product of glycolysis, and further Krebs cycle and electron transport chain, the activity is regulated by ATP. ATP serves as the allosteric inhibitor to many of the glycolytic enzymes. So now, once we have figured out the enzymes, we're going to start talking about getting energy. Now, I know that you all absolutely love the conversations about glycolysis and transition steps in the Krebs cycle and oxidative phosphorylation. I know I'm even more excited than you are. Now, on the serious note, my goal is not to make it overcomplicated. My goal is to give you a conceptual idea and not to bog you down in the 11 reactions of glycolysis. Okay? So instead, we're going to look at major transformations here. But first, I want to write down the equation for aerobic respiration. So look at this. We start with the glucose molecule that has this formula. This molecule is oxidized by oxygen. It's aerobic respiration, producing 6 molecules carbon dioxide, 6 molecules of water, and energy. So what do you think this energy right here is used for? The aerobic respiration, folks. When you hear the word energy, and we're talking about cells, which three letters should pop into your mind? Cell, energy, three letters, huh? ATP. So this energy is used for the ATP synthesis. Sounds good? Okay. So the whole aerobic respiration has three main steps. It's glycolysis, Krebs cycle, and oxidative phosphorylation. We're going to start with the glycolysis. And our main goal will be to count the number of carbons in each intermediate. We're going to start the glucose. So the molecule of glucose, how many carbons does it have, folks? 6. So we start with 6 carbons. During the first 5 or 6 reactions, I cannot remember from top of my head, we use 2 molecules of ATP, so minus 2 ATP, to produce 2 molecules of the intermediate that we're going to call glyceraldehyde 3 phosphate, or G3P. How many carbons does each molecule of G3P have? 3, but we have 2, so still 6 carbons. Now, this investment now starts bringing us the fruits. When this 3 carbon intermediates enter the energy payoff phase, in the series of reactions, they produce a total of 4 molecules of ATP and 2 molecules of NADH. These NADH molecules, do you remember what they carry? So again, excellent. Electrons. So we got ourselves some electrons. We also get 2 molecules of pyruvate. Now, I understand that the question is how many carbons are in each pyruvate. This question is silly. You can count. Okay? So each molecule contains 3 carbons. So we start glycolysis with 6 carbons. We end glycolysis with 6 carbons. And the net yield will be, first of all, how many molecules of ATP net? 2. Hear me out. We invested 2 in the beginning, right? We produced 4 at the end. So plus 4 minus 2. So we get 2 ATPs, 2 NADH, 2 pyruvates. Did I ever mention the word oxygen when I was talking about glycolysis? I did not. Glycolysis is an anaerobic process. Okay? Now, in prokaryotes, let's be real, it happens in the cytoplasm. In eukaryotes, it also happens in the cytoplasm. That makes sense? And this process is conserved, present, in virtually all organisms, all cells that exist, from archaeal and bacterial to mammalian. I just wonder, what does it tell you? That, huh? No. What I'm saying is, that's not, my question is, all cells, no matter how complex or simple they are, they use glycolysis. Evolutionary, what can you say about that process? Yes, it's always been there. It is old. Glycolysis is old. It's probably the first process that arose in heterotrophs to generate ATP. Okay? Good so far? So despite the fact that glycolysis is anaerobic, if oxygen is present, pyruvate molecules can enter the next steps, or step should I say, which is a transition step. So the transition step and the Krebs cycle occur in mitochondria in eukaryotes. In prokaryotic cells, such as bacteria and archaea, these processes still happen in the cytoplasm. So let's take a look at the fate of the pyruvate here. These are carbons of the pyruvate molecule. There are 3 of them. So when pyruvate is oxidized to a molecule called acetyl-CoA. So this is the acetyl part, which is derived from the pyruvate, and this is a coenzyme A, which is a cofactor in this reaction, so it comes and goes. So if you look at the acetyl part, how many carbons does it have? So we went from 3 carbons to 2 carbons. That missing carbon, where did it go? Carbon dioxide, yes. And this oxidation, this decarboxylation of pyruvate also generated a molecule of NADH for you. Okay, so now you have this acetyl-CoA with 2 carbons. And this 2-carbon intermediate enters the Krebs cycle, also known as citric acid cycle or TCA cycle, where TCA stands for tricarboxylic acid. Now, terminology is great, but let's take a look at the concept here. So we have 2 carbons in acetyl-CoA that are derived from the original glucose, and 4 carbons in the molecule of oxaloacetate, which is one of the intermediates in the Krebs cycle. I'm going to ask you a bunch of very silly questions. What is 2 plus 4? 6. So the combination of these 2 molecules results in the formation of the citrate, the 6-carbon intermediate of the cycle that first goes through the isomerization to isocitrate, and isocitrate loses carbon dioxide and goes from 6 to 5 carbons in the molecule of alpha-ketoglutarate. Okay? So this process, that oxidation of isocitrate, forms an extra NADH. alpha-ketoglutarate is further oxidized, losing another carbon dioxide and yielding another molecule of NADH, producing succinyl-CoA, 4-carbon intermediate. Succinyl-CoA turns into the succinate, producing ATP. Now, let me give you full disclosure in terms of what you need to understand. I don't want to focus my questions, I'm not going to focus my questions on you memorizing each individual step of the Krebs cycle. I want you to understand, like if I ask you when citrate, 6-carbon, is converted to alpha-ketoglutarate, 5-carbon, what happens to that lost carbon? Becomes CO2. Okay? So I want you to, because we're gonna go back to this loss of carbons, and we're gonna discuss why this is important. What does it mean conceptually to our reaction that we wrote before when glucose is oxidized to CO2 in water? The point is, this 4-carbon intermediate then gets a bunch of conversions that do not include the loss of carbon, okay, but yield oxaloacetate at the end, so the cycle can continue. During these conversions, what is produced? ATP, as we mentioned before, FADH2, which carries what? Electrons, yes, and another molecule of NADH. Now, let's take a look at this. Again, I'm going to erase all of this. And I want you in your head to return to glycolysis and tell me something. At the end of glycolysis, how many pyruvates do we have? 2. So the processes that we just went through, the transition step, the Krebs cycle, in other words, like this page, for 1 molecule of glucose, we have to run it twice. Would you agree with me? Yeah, because we need to process 1 pyruvate and then another pyruvate. Does that make sense? What I'm trying to convey, yes? Yes? So what we went through was for 1 pyruvate. Glucose catabolism during glycolysis gives you 2 of them. So you need to do 2 transition steps and 2 Krebs cycles. Okay? I will not move on until I have some kind of a confirmation from every person in this room that every person in this room understands what I'm talking about, and if you don't, let me know right now. I cannot read your minds, thankfully. So I would appreciate a nod, or yes, or I have no idea what you're talking about. Yes. Perfect, thank you. So I'm going to ask you questions. This is 1 molecule of glucose, right? Right? So how many pyruvates do we get in the end? 2. So we're going to run a transition reaction for 1 pyruvate and then for another. So we can multiply products by 2. And then, since we have double the products, we need to multiply the Krebs cycle by 2. Excellent. So now let's figure out what we get out of this. Let's count the number of carbon dioxide molecules. I deliberately keep the entire picture in the frame. 1, 2, and 3. Right? So, altogether, for the glucose molecule, what do we need to do with the number 3? We need to double it, right? So, per molecule of glucose, transition step in the Krebs cycle gives us 6 molecules of CO2. Okay? Now what we're going to do, we're going to do the same for NADH. 1, 2, 3, and 4. So what we have to do with this number 4? We have to double it. So 8 NADH molecules. The rest is, well, fairly easy. We need to count ATP. There's only 1, so we need to double it. Therefore, 2 ATP. And finally, FADH2. There's only 1, so 2 FADH2. So a couple of comments. So first of all, how many carbons in the original glucose molecules do we have? 6. How many carbon dioxide molecules do we have at the end of the Krebs cycle? Also 6. By the end of Krebs cycle, we're done oxidizing carbons from glucose. All carbons from the original glucose are oxidized to CO2. Okay? Second, just be honest. It was a pretty long and winding road. Does the yield of, 2 molecules of ATP, does it seem impressive? It doesn't. So we went through a hell of biochemical reactions with glycolysis and transition step and Krebs cycle. And so far we managed to snatch 4 molecules of ATP total, 2 in glycolysis and 2 in Krebs. Quite obvious that these processes are not for ATP. These processes are to generate a buttload of NADH. In other words, we accumulated a huge number of high-energy electrons that are sitting on those electron carriers. So now we're going to bring those electron carriers into the electron transport chain. So this is it. This is your NADH. Remember, I compared it to a taxi. Like it carries, and if I didn't, I'm going to do it now. NADH is an electron taxi. When you get into a taxi, you are expected to get out of the car at the end of the trip. Right? You're not staying there forever. Same is true for electrons and NADH. They're not staying there forever. So, NADH, when it encounters the proteins of electron transport chain, it loses its electrons and becomes NAD+. When a molecule loses electrons, is it oxidized or reduced? So NADH is oxidized. And the complex 1 of the electron transport chain, which receives those electrons, is it oxidized or reduced? Reduced. Okay? FADH2 faces pretty much the very similar fate with the only difference that this molecule gives its electrons to the complex 2, the electron transport chain. Now you can see that these molecules, the electron transport chain is located in the membrane, right? Okay. What is this membrane? If we're talking about eukaryotes, it is the inner membrane of mitochondria. But what about prokaryotes, like bacteria? Do they have mitochondria in the cell? They do not. Say again? Nope. It's still in the membrane. What membrane do they have? The cell membrane, yes. It is the cell membrane in bacteria, for example. So we're going to consider this, out and in the cell, okay? But conceptually, the process isn't different. So here what happens. These electrons enter the first complex, or second, whatever, and they start, you can trace this arrow, they start moving from one protein, the complex, to another. Okay? I usually compare it to a ball that is pushed off of the top, the flight of stairs. So if this is the flight of stairs, and you put the ball right here, and you push it, with every fall, it will release some kinetic energy, right? Yeah? So it's not kinetic energy, but the idea is pretty similar. With every electron transfer from one complex into another, the energy from those high energy electrons is released. Okay? So what this energy is used for? It is used to pump hydrogens, so that's a pump hydrogens from one side of the membrane to another. Now when you look at this part, this side, like this, how would you compare the concentration of hydrogens on top of the membrane to the concentration at the bottom? On top, it's bigger, right? So we have big hydrogen concentration here and small hydrogen concentration here. So we have a chemical gradient. Would you agree with me? What about charges? The top is what? Compared to the bottom? More positive. So we also have gradient of charges. The electrical gradient. So overall, this creates an electrochemical gradient which is supposed to drive positively charged hydrogen ions back. Can hydrogen ions cross the membrane directly? Why can't they? Because of the hydrophobic tails of these guys? What about hydrogen ions? Are they hydrophobic or hydrophilic? They're hydrophilic because they charged. Make sense? So in order to cross the membrane, they have to use a channel. This channel right here is the part of a protein called ATP synthase. So hydrogen ions diffuse. This is the diffusion. Can you consider diffusion a movement? Sure. Yeah, it's a movement. Movement entails kinetic energy. So essentially, the energy of moving hydrogen ions is what's used for the synthesis of ATP from ATP and phosphate. Okay? So I developed, and probably I am compensating for the B in my middle school art class, I've developed a pretty decent analogy that illustrates and puts it all together a little bit better and becomes less abstract. You're all familiar with the concept of a water mill, right? That was used before, we had like electric power and stuff. So imagine that we have a water mill, which is ATP synthase. So when it rotates, it churns out ATP. By the way, just FYI, that ATP synthase on the left, this enzyme quite literally rotates. It makes turns, and per each full turn, it produces 3 molecules of ATP. It was established, like it was observed. Okay? So let's go back to this. In order to function, water mill has to have water falling onto it, right? So this is water. This water represents hydrogen ion diffusion. So this water comes out of the pipe. Now, the flow of this analogy, I have no name for the pipe. It's just a pipe, okay? But the point is, this pipe goes through the ceiling, which is a membrane. But in order to have these hydrogen ions up, you have to pump them. So far, do you follow? So you have a little pump. The pump are the complexes 1, 3, and 4. These complexes that you see that pump hydrogen across the membrane. Okay? Now, what provides energy for pumping? Nope. Electrons. That's it. You don't need to add anything. Electrons provide energy for pumping. So right here, we have a little, like a gas tank, okay, with electrons. This is how you fuel your pump. Does that make sense? And these electrons are brought there by the taxis, NADH and FADH2, and the electrons are taken away from from glucose during glycolysis and Krebs cycle. Does that make sense? Okay, so yes. And what do you think NAD plus does next? Yeah, it's a taxi. It goes back. It goes back to pick up more electrons, just like a taxi goes back to pick up more passengers. Yes, and this is an often overlooked idea. Not the concept in this. You have to unload a taxi. Does that make sense? You have to get rid of those electrons from NADH so you have more available NAD plus to carry more electrons. Does that make sense? Well, this is the summary of aerobic respiration, okay? I'm not going to recite the entire table. As a matter of fact, I'm not going to recite these two columns because we already discussed this. Now, few things. First of all, some differences. So you need to know the products of each step. Okay? Now, for glycolysis, as we mentioned, it's worth mentioning again, no oxygen is required for these two steps. In order for them to run, oxygen is required. You're not going to run without oxygen, and we're gonna figure out why. But for now, does that make sense? So it turns out that the energy of electrons that are sitting on one molecule of NADH is sufficient to make 3 molecules of ATP. So it's not really an equivalency, but we can do this. NADH equals 3 ATP. The energy from the molecule of FADH2 will give you 2 molecules of ATP. Okay? Good? So I'm gonna skip the boring arithmetic. I can tell you all these calculations are correct. So at the end of the day, the maximum possible theoretical yield, and I can't emphasize it enough, theoretical yield of aerobic respiration is 38 molecules of ATP. Am I clear? So I have one question, big question, which is not, it's kind of like 3 out of 10 on the difficulty scale. Why does NADH gives you 3 ATPs in the end, and FADH2 gives you only 2? The answer is on the screen. Go on.
Is it because FADH uses its electrons to actually pump out the hydrogen?
No. What about the complex 2? The complex 1? Yeah. So in case of FADH2, you start on the lower step. That's it. The electrons manage to go through, less number of proteins in the electron transport chain, and that brings, less molecule, less ATP, less hydrogens across the membrane. Does that make sense? Okay. Now I have a question which is like 8 out of 10. It's totally fine if you don't answer that. So in reality, the yield here doesn't even reach 30. It's usually slightly less than 30. Can you suggest why? It's a very hard question, seriously. Well, yes. The transport, first, great answer. The transport in eukaryotes, the transport of pyruvate into mitochondria requires ATP, yes. But that's not the main contributor. Okay, I'm giving you a hint. Which process is directly responsible? We're going to go step by step. Which process is directly responsible for the energy for ATP synthesis? Directly. Huh? No. Directly, like ATP. Which protein synthesizes it? ATP synthase. What provides immediate energy of that synthesis? No, not electrons, not ADP. Say again? Diffusion. Why do hydrogens use that molecule, ATP synthase? Because they what? Hydrophilic, so they can't cross the membrane. At all? Some of them manage to pass it by. Membrane is a little bit leaky for hydrogens. So some of the hydrogens that are pumped across the membrane bypass the ATP synthase and are essentially useless. So both this and the use of some ATP in the entire process contributes to the fact that it's not 38, it's less than that. Still pretty good. Okay? We have one problem that we haven't resolved yet. It's electrons. So at the very end of the electron transport chain, we have electrons. And they cannot move around on their own. Does that make sense? They can't just hang out. We have to drop them on something. That something is called final electron acceptor, and in the case of aerobic respiration, it's oxygen. Oxygen combines with 2 electrons and 2 hydrogens, forming water. Yeah. That makes sense? Yeah? Say again? What do you mean by leftover?
Okay, you're saying so we have extra electrons.
It's not extra, we just have electrons. They have to be attached to something. And this something is the final electron acceptor, which in case of aerobic respiration is oxygen. Are you with me so far?
So are you saying the hydrogens, because of the leaky membrane, come through, and then those hydrogens become water?
Not necessarily those, but some hydrogens. There's no distinction. They all are identical. Now, yeah, so just, we have some surplus of hydrogens that we can use. So, as we just mentioned, aerobic respiration uses oxygen as the final electron acceptor. Can we use some other molecule? The answer is yes. We can use an inorganic compound, such as sulfate or nitrate. So you can see this illustration of anaerobic respiration final steps in Pseudomonas stutzeri. If you wonder why the spelling is weird, is because this entire illustration in Portuguese, it is the only one that was not copyrighted that I could find. Okay? So basically, here's the idea. You see those electrons moving around? Eventually, they get accepted by nitrate, and it can stop at nitrite, or it can produce nitric oxide, or it can produce nitrogen at the end. Not the point. The point is that the final electron acceptor in anaerobic respiration is an inorganic molecule that is not oxygen. Am I clear? Okay? So, if you're concerned, am I going to ask about this in the exam? Probably, yes. But if you're concerned about the formulas and or the names, don't worry, because if you need a formula based on the name, I will provide it. If you need a name based on the formula, I will provide it. Not a problem. I want you to conceptually understand that in anaerobic respiration, at the end of electron transport chain, is not oxygen, just a different compound. Okay? I'm going to ask you questions about aerobic respiration first. They are very simple. We answered them gazillion times today. Aerobic respiration, does it include glycolysis? Yes. Does it include Krebs cycle? Yes. Does it include electron transport chain? Yes. Anaerobic respiration, does it include glycolysis? Yes. Krebs cycle? Yes. Electron transport chain? Also yes, but it's different. So the difference is what's at the end. Does it make sense? The whole difference is what is at the end. It's same old glycolysis and Krebs, but electron transport chain is what's different. Does that make sense? For instance, species of Pseudomonas, they can do both. They can respire aerobically or they can respire anaerobically. Okay? Sounds good?
So in the Krebs cycle, if it's anaerobic, what would be the product?
It's not going to happen. Krebs cycle cannot run without oxygen or without that inorganic compound. Think about this. All the taxis are full of electrons. And if there is nowhere to dump them, it will automatically inhibit Krebs cycle. If you get a shitload of NADH and it doesn't have the functional electron transport chain to give electrons to, it will shut down the Krebs. Now, if you give me 3 minutes, you will see where I'm going with this. So far, aerobic versus anaerobic respiration, does it make sense? Yes? Now, anaerobic respiration will always produce less ATP than aerobic. I'm not going to go into the chemical reasons for that. I want you to think this way. So if we are looking at the energy difference in case of aerobic respiration, there is glucose, and it gives its electrons to oxygen. This distance represents the amount of energy, the amount of ATP that can be produced. In anaerobic respiration, glucose gives its electrons, for example, to nitrate. You see that this span is much shorter. This is true for nitrate, sulfate, doesn't matter. Okay, oxygen is the most efficient final electron acceptor. We good so far? So what's going to happen? There's no oxygen. We still have to unload. So if there is no oxygen, cells, if they can, will switch to fermentation. So 2 types of fermentation that I want you to know are lactic fermentation and alcoholic fermentation. So on the right, this is lactic fermentation. Okay? So glucose, in the process of glycolysis, is converted to 2 pyruvates. Any questions? It also generates 2 molecules of NADH that carry electrons. You need to free them up. Right? Right? You need to free them up. So these NADH molecules unload the electrons. Listen to me very carefully now, because I'm going to ask you a question about what I just said. They unload the electrons, reducing pyruvate. So it becomes lactate. What is the final electron acceptor in lactic fermentation? I'm going to say it again. NADH unloads its electrons, reducing pyruvate. So it becomes lactate. What's the final electron acceptor?
Oxygen.
There's no oxygen, huh? Come on. Dude. No. Glucose is long gone. Glucose is gone. We converted glucose into pyruvate. No. No, okay. So this is a pyruvate. This is NADH.
Right.
So NADH loses its electrons. No. It loses its electrons, becoming NAD. Okay, fine. Pyruvate becomes reduced. What is reduction? Getting electrons. So what is the final electron acceptor? Pyruvate, yes. Pyruvate in this case is the final electron acceptor. Does that make sense to you? So NADH loses its electrons, pyruvate gains them, becoming lactate. No oxygen is involved. It's not required. And the final electron acceptor is some kind of organic compound. Now, since you are equipped with that knowledge right now, let's take a look at alcoholic fermentation. So you have pyruvate. The first step is it's losing carbon dioxide, forming acetaldehyde. NADH loses its electrons, reducing acetaldehyde to ethanol. What is the final electron acceptor here? No, not acetaldehyde, because this is acetaldehyde that receives those electrons. Again, organic compound. So in any fermentation process, oxygen is not essential, and the final electron acceptor is the organic compounds. So when we say that fermentation is anaerobic, it doesn't mean that it must not have oxygen. It means that it doesn't care about oxygen. Am I clear? Do you understand what I'm trying to say? You can ferment things in the presence of oxygen. Oxygen is just, it's not needed. Okay? We're going to talk more about this. I'm going to very quickly go back and highlight this. When organisms respire, they produce free radicals or reactive oxygen species. One of them is hydrogen peroxide. What do you use hydrogen peroxide for at home? Yeah, killing bacteria, right? So if bacteria produce hydrogen peroxide, they should be able to neutralize it. That's poison for them. Some cells can't. So this is the reason why they can't respire. They don't have the necessary genes. Okay? That makes sense? Let's go back to fermentation and just, we have very few things left to talk about, but obviously don't memorize the table. I just want you to acknowledge and appreciate the massive role that bacterial fermentation plays in our lives. Okay, this is like, when you fuel up your car and the gas that you pour in the tank contains 10% ethanol, do you know where this ethanol comes from? Corn. This is what corn in the United States is grown for the most part. It's cattle feed, sure, human feed, sure, but most of the corn is used for ethanol production. In fact, in Brazil, practically all cars run on ethanol because they have a shitload of sugarcane, which they use for ethanol production. It's one of the biggest producers of ethanol. Nail polish remover. There you go. Fermentation. Okay? Obviously, delicious things like beer and wine. That's fermentation. Bread. Sourdough bread. Lactic fermentation gives us various types of fermented cabbage, kimchi and sauerkraut, as well as dairy products. But this is my favorite, actually. I don't remember if I talked about it. But if I didn't, one of the goals that I set for myself in this course is to develop in you the aversion to as many food products as possible. As you may know, there's a type of cheese that is produced in Switzerland that is very, very smelly. Okay? People sometimes compare it to the smell of well-worn, aged socks. It's fine. I tried it. It's good. The pretty rancid, but edible. Its smell can be attributed, for the most part, to a genus of bacteria called Propionibacterium. Interestingly, human skin has Propionibacterium on it as a normal component of our skin microbiome. It's called Propionibacterium acnes. It houses in our hair follicles and feeds on the oily secretion of our sebaceous glands. And when we enter puberty, we start producing a lot of that oily secretion, and this bacterium, Propionibacterium acnes, starts growing like crazy, often, which may cause inflammation and result in acne. Okay? Now, if you wonder, what is the connection between the acne and the Swiss cheese? If somebody has a zit, and they would pop the zit and spread that white substance between their fingers, it's going to smell like Swiss cheese. This is the reason why Swiss cheese and socks share the smell, same metabolites. Okay? Absolutely, the same chemical compound that gives the smell of the socks, it gives the smell, similar smell to Swiss cheese. Okay? Now, to lighten you up, after the Swiss cheese, let's talk about Streptococci. So the reason why I turned to strep is it's a fermenter. It's a classic fermenting bacteria, okay? So the first species I wanted to mention is Streptococcus pyogenes, which you all know is a causative agent of strep throat. Bacterial pharyngitis. What do you do now if you have strep throat? Yeah, you go get antibiotics. What if you don't? There are some estimates that in late 19th century, before the invention of antibiotics, about 80% of children who ended up with streptococcal infection of the throat died. No, it's an extent, no. But if you read you read a biography of some president of the U.S. in 19th century, and they say, they bear 7 children and 4 of them died of fever. It's no fever. It's most likely strep throat. So if you don't treat it, it becomes systemic, leading to scarlet fever, which is pretty severe disease in itself, and it may aggravate itself further, resulting in rheumatic fever. In rheumatic fever, Streptococcus infects cardiac valves, causing endocarditis, and joints, therefore rheumatic. Endocarditis results in the heart failure and death. Okay? Does that make sense? That's pretty bad. So this is why we focus on antibiotics. But that is not the only thing that it can cause. Because just like Staphylococcus, it can cause a variety of skin infections. So impetigo, erysipelas, cellulitis, erythema, all of this can be caused by streptococcal infection in the skin or subcutaneous tissue. Okay, now, it's not such a huge problem because we can treat it with antibiotics. Okay? The transmission, obviously respiratory, and the contact. And interestingly enough, people can carry it asymptomatically. So somebody can carry strep pyogenes and not being sick at all. Okay. Now, strep mutans causes the most common bacterial disease in the world, which is dental caries. So it's present, can be present in the oral microbiome, and it thrives in the sweets. So it uses glucose to form a sticky substance to, well, stick to the surface of the teeth, and ferments fructose, producing acid. And this acid causes the decay of enamel and then dentin and, eventually pulpitis. So, full disclaimer, when you brush your teeth, you don't remove it. You just wreak havoc, like it's all messed up. So it tries to rebuild. By the end of the day, you wreck it again, okay? But glucose and fructose are both monosaccharides. And when you combine them, which disaccharide would they form? The answer is on the screen. Like, I'm going to bring it here. Sucrose, which is sugar. So when you eat or drink products with a high sugar content, you feed your strep mutants. Does that make sense? Yeah, so like regular Coke or any sweets that are sweet because of the sucrose, that works towards that. Okay? Make sense? Okay. So a couple more. Strep pneumonia, you will hear about that if you work with pediatric or geriatric populations. Streptococcus pneumonia causes community-acquired meningitis. Quick question, not for the exam, but just so we are on the same page. How do you understand the term community-acquired? So, okay, nursing home.
It's being spread by somebody who you regularly come into contact with.
Yeah, like a school, right? Nursing home. Gym, sure. But if you go, like, let's say you work at a hospital. If you get something in a hospital, that's hospital acquired. I know that the line is pretty fuzzy, but it's hospital acquired. We try to distinguish because there are sick people in the hospital. Like, when you go in the hospital, there are sick people there. When you go to school, you presume that people are fairly healthy, okay? So the transmission is contact and respiratory. Now, the reason why I mentioned geriatric and pediatric populations, those populations usually crowded either in a nursing home or a school, and they have terrible hygienic habits. So if you're familiar with this, have you heard the term or the brand, I don't know how to call it, Prevnar? Harper? Yeah? Yeah. Well, Prevnar is a vaccine against pneumococcal meningitis. It is mandatory for kids who go into public school in the state of Ohio, along with Hib and meningococcal vaccines, so 3 meningitis vaccines, and it is mandatory for people who get to live in the nursing home. Okay? Because it's bad if it goes. It obviously can also cause pneumonia, but Streptococcus pneumonia, but we're concerned about meningitis. Now, this bug, Strep A galactiae, also known as Group B Streptococcus, doesn't ring a bell? Anyone? It might. Especially if you have kids. So Group B Streptococcus, A galactiae, is found in the microbiome. can be found in the genital microbiome, but also in the GI. It is absolutely fine for a woman, but it can cause neonatal meningitis. It infects newborns when they pass through the birth canal. This is why before the labor, before the delivery, women are tested. Because if it is present at a certain level, there will be a C-section. Nobody's going to risk it. Okay? Does that make sense? And it's an example how a microorganism that is benign in its regular location can become a killer in a different environment, such as meninges, such as the brain. Okay? Good? So we have a couple more things to talk about. This is going to be a short conversation, I promise. So obviously, carbohydrates is a great source of energy. Okay, right here. We break it down to glucose, and I'm not going to repeat the whole story again. You get the gist, right? This is what we were talking about for the last hour. What if there's no glucose? Is there another energy source? Yes. Fats, triglycerides. If you break down triglycerides, they form glycerol and fatty acids. Glycerol can be converted into G3P, and you can see where it goes next. Fatty acids, through the process of beta oxidation, can be converted to acetyl-CoA. All clear? What if you only have proteins available? Proteins will be broken down to amino acids. Amino acids go through two processes. You just need to know the names. It's deamination and transamination of amino acids. These processes can either yield pyruvate or can produce the intermediates of the Krebs cycle. Does it make sense to you all? But the biggest take-home message from this is that these arrows actually should go in both directions. This is called amphibolism. Amphi stands for both, too, okay? So what does that mean? It means that the same way you can break down amino acids to yield intermediates in the Krebs cycle, the same way you can use intermediates of the Krebs cycle to make amino acids or to make fatty acids. Do you follow? So, like, seriously, this is it. The information that I gave you is enough. Which brings us to photosynthesis, which is a fascinating process, first of all. It's really cool. But also, it kind of gives me a chance to take a look at the bigger picture in terms of energy process. So, real quick, I'm going to write the reaction for you. What is that? Which process is that? Say again? No. What? Aerobic respiration, right? Aerobic respiration. Glucose is oxidized by oxygen, producing carbon dioxide, water, and energy. If you run this reaction backwards, carbon dioxide and water are combined with energy to produce glucose and oxygen. Backwards, It is photosynthesis. Cool? So, where does this energy come from? Light. It comes from light. So, photosynthesis happens in eukaryotes on the membranes of structures called thylakoids, which are confined within a distinctive organelle named how? Chloroplast, yes. So these stacks of thylakoids are in chloroplasts. These chloroplasts contain photosystems, and one of the key elements of each photosystem are pigments that absorb light. Now, obviously, I will definitely ask you to memorize all the colors of all the pigments that I present on this slide. Obviously not, but it's color-coded. My goal here is to show you that photopigments span the entire spectrum. Question? Span the entire spectrum of visible light, which makes bacteria very versatile in the utilization of that light. Some of them are going to thrive in the morning, in the evening, this light, that light, depth, whatever. Does that make sense? Because each pigment absorbs a specific range. And the reaction of photosynthesis, they can be divided into two main categories, conceptual categories, which are light dependent, that obviously require light, and light independent. So what I want you to process, light dependent reactions, hear me out. The goal is to harness the energy of light to produce ATP, which is energy source, and NADPH, which is an electron carrier, to generate some high energy electrons. These electrons and ATP are then used to convert inorganic molecule of carbon dioxide into organic molecules that we call carbon fixation. So let's take a closer look at how this works. So we're going to start with the light. This light is absorbed by a bunch of harvesting centers, whatever. My point is, does light have energy? So this energy is transferred onto the reaction center, and this energy is used to, figuratively speaking, give a massive push, a kick in the butt to an electron. Okay? So this light energy is transferred to an electron. And this high energy electron, shown here, enters the electron transport chain. I'm not going to say a word about electron transport chain. We spoke about it enough. The goal, to generate hydrogen, the electrochemical gradient, for eventually the ATP synthesis. So this process is mediated by photosystem 2. So photosystem 2 transfers the light energy to an electron. Electron enters the electron transport chain. ATP is synthesized because of the hydrogen gradient, right? So here you see an electron pretty bummed out at the end of the chain. When it enters photosystem 1, I think the numbering is because of the order of discovery, where it receives another jolt of light energy, and that, well, makes it high energy, and the electron is accepted by NADP+, forming NADPH. Sounds good? So it's the same electron. It goes through like bouts, okay? Like it is energized. This energy is used for proton gradient, which is used for ATP synthesis. Then it's energized again, and it gets attached to NADP, forming NADPH. Am I making sense? Yeah? Yeah? Right. So there's one little problem. There's a little hiccup here. This. We need to recover the electron. So we need an electron donor. It's water. So water loses an electron. Can we say that the water is oxidized? Yeah. It loses the electron. Yes. So water loses the electron. It is oxidized, okay? More specifically, it's oxygen within the water that's oxidized. And this is why, in addition to glucose, as we'll see, another product of photosynthesis is oxygen. But does it have to be oxygen? The answer is no. So this is oxygenic photosynthesis, the one that produces oxygen. But if we replace water with some other donor of electrons, let's say it's going to be hydrogen sulfide, it's going to be fine. The sulfur will be produced instead of oxygen. As a matter of fact, a lot of sulfur, pure sulfur that is mined in the world, is essentially the result. It is a product of anoxygenic photosynthesis that happened like in the ocean a gazillion years ago. Okay? Do you see the difference between oxygenic and non-oxygenic? You just need a non-water donor of electrons. But what do we use this ATP and NADPH for? So here we go into light-independent reactions. And I know this Calvin-Benson cycle looks fairly intimidating. But let's just do arithmetic. How many carbons are in this molecule? 5. So if we take 3 molecules like this, 3 molecules of ribulose biphosphate, we get 15 carbons altogether. Do you agree with me? How many carbons are in 3 molecules of carbon dioxide? 3. 15 plus 3 gives you 18. So how many carbons are in 6 molecules of phosphoglyceric acid? 18. There you go. For God's sakes. So 18 carbons. Phosphoglyceric acid is ready to be reduced. So the energy for the reaction comes from ATP. The electrons for the reduction come from NADPH. Right? And what we get in the end is glyceraldehyde 3-phosphate, or that G3P molecule that we talked about before, the intermediate in glycolysis. So we have 6 molecules of 3 carbon, G3P, so 18 carbons total. 1 molecule is being taken out. It will be used in the future to make a molecule of glucose. Fair? Okay, good. So we have 5 molecules left. Each molecule has 3 carbons. 15 carbons that will be rearranged to form RUBP, and the cycle repeats itself. So the main point here is that we take 3 carbons out of inorganic CO2 and generate 3 carbons in organic G3P. When we incorporate any inorganic source into organic compound, we call it fixation. So this Calvin-Benson cycle is carbon fixation. Okay? I'm going to hold you for 5 more minutes to illustrate how that knowledge can be incorporated into the history of life on Earth. Now, full disclaimer, you can doze off. It's not for the exam. But I find it an extremely fascinating topic. So, the Earth was formed roughly 4.5 billion years ago, and the traces of the first life can be dated to about 3.8, 3.9 billion. Okay? So, few questions. Question number 1. The original atmosphere on Earth, did it contain any oxygen? No. Anaerobic atmosphere, right? So no oxygen. Did it have any organic food? No. So first cells. And when I say no, probably there were some organic compounds, but it was like, So first cells, were they autotrophs? Remember, the ones that can convert inorganic carbon into organic. Or heterotrophs, autotrophs. Sorry. Were they aerobic or anaerobic? Now, what protects Earth, like surface, us, from the UV light? Say again? Ozone layer. Does anyone know the formula of ozone? Yes, O3. So it's oxygen, right? Just 3 atoms. So at the very beginning of Earth, did it have ozone layer? No, there's no oxygen. Like there's no way to get it, right? Do follow. So the Earth, the surface, was exposed to pretty heavy bombardment with UV light. So one of the hypotheses about the origins of photosynthesis, it was the process to accumulate and dissipate the energy of sun to protect the cells in the first place, not to make ATP. See what I'm saying? Like, if I'm getting heat, maybe I can use this energy for some use, okay? So eventually, these autotrophs develop and they start running photosynthesis, okay? So what are 2 products of photosynthesis? Water, yeah, water, there's plenty of water, but more importantly, glucose and oxygen. Do you see that? Right here. So glucose, fine, it's a food, okay? What about oxygen? What's going on with that oxygen? So oxygen, which is the product of photosynthesis, can oxidize the living shit out of pretty much anything, right? Sorry. Oxygen oxidizes everything. And there are plenty of things to oxidize. So those anaerobes tirelessly speed out oxygen. This oxygen tirelessly oxidizes whatever's available. Therefore, the atmosphere remains anaerobic. At some point, the Earth runs out of things to oxidize. So oxygen isn't used in those reactions. What starts happening with the levels of oxygen in the atmosphere? They start increasing until so-called Great Oxygen Revolution, or sometimes I prefer to refer it as Great Oxygen Catastrophe. These guys are strictly anaerobic. So they pollute their own environment with oxygen so much that, according to the paleontological estimates, 90% of life, not species, life on Earth dies. Gone completely. It was the greatest extinction in the history of Earth. Dinosaurs was a child joke, to be honest, compared to this, okay? What's left? Organisms that can survive in the presence of oxygen. I mean, there are some anaerobes, strict anaerobes that are left in some nooks and crevices, but aerobic metabolism is much more efficient, and since then, aerobic organisms dominated. And if you think about this, it's kind of unfair, because they came to the prepared playing field and took it over. Okay? So my point is, if you know the biochemistry and, of the microorganisms, you can actually explain the events that happened billion years ago. Okay? Good? Questions? Okay, let's wrap it up.